Now we proceed to the second lecture that is going to be on the Nobel Prize 2020 in the field of chemistry. We have Injara to introduce us to the speaker, Dr. P. Chandrashekar, Center for Cellular and Molecular Biology, Hyderabad. Over to you, Injara.
Good afternoon, all. I am Inchara from 5th semester Molecular Biology. It is now my great pleasure to introduce Dr. P. Chandrashekar, our speaker who will talk about development of a method for genome editing. Dr. P. Chandrashekar graduated from Bangalore University with a Masters of Science degree in Biotechnology in 2002. He received his PhD in stem cell biology, RNAi, and mouse development from the Center for Cellular and Molecular Biology, Jawaharlal Nehru University in 2010. He completed his postdoctoral fellow on stem cell biology and mouse development from Mount Sinai School of Medicine, New York, USA, on October 2010. He is currently the senior scientist and a Wellcome Trust at DBT India Alliance, Early Career Fellow at Center for Cellular and Molecular Biology, Hyderabad. Sir, we look forward to your talk about genome editing. Over to you, sir.
Okay, thank you very much for the introduction and also thanks for the invitation. You guys can hear me?
Yes, sir, we can hear you.
Okay,
Yes, sir.
share my...
Yeah, you can start presenting, sir.
Yeah. So are you able to see my screen?
Yes, sir, we can see.
Okay. Okay, thanks a lot for the opportunity. And okay, so my lab basically works on stem cells and mouse genetics, but we use CRISPRs for editing the genome of the mouse stem cells as well as mouse. So that's how I'm here to talk to you on this. So the first slide is self-explanatory. This is something I picked up directly from the Nobel website, where you will see genetic scissors. The title is The Genetic Scissors, a tool for rewriting the code of life. And as we know, the title of the Nobel Prize was for development of a method for genome editing. Now, this was awarded to two ladies. This is the first time that has happened in the history of Nobel, that both are women. And one was Emmanuelle Charpentier from Max Planck Unit of Science and Pathogens. She's the director of Max Planck, one of the Max Planck institutes now. And another is across the Atlantic, that is from University of California, Jennifer Doudna. So these two were given the Nobel Prize for defining the mechanism, how the CRISPR system, which we are going to look in the future slides, works and how it can be utilized. So most of the Nobel Prizes, as the previous speaker said, takes a lot of time to be awarded, but there are few which are exceptional and their impact is so fast and profound that these get Nobel Awards very quickly. And in this case, it's 8 years, and RNA interference was one more, and I think the other one was induced pluripotent stem cells for Yamanaka, which came very quick. Okay, so the impact of this technology is actually game changer for biology. What I'm going to do is, throughout the talk, I have divided into two parts. One is, in the beginning, I will talk to you about the history of how it originated and how different research groups contributed different level of findings and information for evolution of understanding the CRISPRs, and ultimately the mechanism was described or discovered, and later on its application was shown in many organisms. So one is the history. Second is, since I realize that most of you are either graduate or master students, so I would talk to you about how it exactly works, the mechanism of it, and how in the past 8 years it has been utilized. I will give you only a few examples. In biology in particular, I'm not going to talk much on its applications, although it's also commercially exploited in many of the areas of industry, biotech industry. That I am not going to. Okay, so this journey also started, the CRISPR journey also started way back in 1987. I'll keep telling you about how things started, and probably you may be able to imagine and appreciate how science evolves from something that is not known at all to a proper understanding how things work. So the first paper, anything got to do with CRISPRs, was not on either genome editing or anything. It was from a Japanese group. The first author is Ishinu. It was in Journal of Bacteriology way back in 1987. They were trying to study the nucleotide sequences around a gene called IAP. You can see on the left, I have marked it. And what they found, in those days, the sequencing was also very primitive. When they tried to do the sequencing by running long gels, and what they found is, I have marked it in the green block here, they found an unusual structure. An unusual structure was found in the 3' end of flanking the IAP gene, and it had 5 homologous sequences of 25 and 9 nucleotides, which were direct repeats, okay, spaced by a 32 nucleotide spacing. Mean to say, they were studying this gene, IAP gene, and when they were sequencing the DNA around it, what they found was, on the 3' end of this gene, they found a small repeat sequences, 5 repeat sequences of the same sequence, and they were interspaced by some 32 nucleotides. So this was little interesting find, but they don't know what they are, what they do. They only notice that there is something interesting, that there are repeat sequences available. So that is the first thing they saw. And they just left it there because that was not their interest to study what these repeats were. And after 5 years, on the right you can see a similar repeat sequence was seen in Archaea. The first one was in E. coli. So some other group found such repeat sequences, not the exact repeats, but different. But the point to note here is they are again repeat sequences of couple of bases, repeated couple of times. They were found in Archaea. Later on, during the 90s, this sort of repeats were observed in multiple bacteria in Archaea. So people started noticing them. They knew that such repeat sequences were there, and whoever noticed, they reported them, but they did not know what these were, what they did, and what they stand for. Then the next breakthrough came up in 2002. So the whole decade went in just reporting such things. On the left, I have put a mark here, the red box here. This was the first paper to name them as CRISPRs. And this is a bioinformatics paper. So these guys identified, they were analyzing all the sequences. And what they observed was, in most of the species, they could see that there were some repeat DNA, which were in couple of repeats. The repeat numbers may vary, but they were always found in a cluster at a single locus. And they were also associated with some enzymes there, some proteins, Cas proteins or Cas genes, the genes which can code for certain proteins, mostly the nuclease-like proteins. Okay, so that is where they, since they were seeing a cluster of repeat elements, that's why they named it as CRISPRs. Sorry. So yeah. These are clustered, regularly interspaced. So it's a cluster of regularly interspaced short palindromic repeats. So they were talking about these repeats which were observed there. And they were also associated with genes called as Cas genes, which are mostly by bioinformatics look like nucleases, which can cut DNA or RNA. Then later on the right, if you see, 3 papers came back to back from different groups in different journals, which showed that these CRISPRs showed characteristic features. The repeat sequences in this showed characteristic features of extrachromosomal origin or coming from some other elements, foreign genetic elements like bacteriophage. You know, for the bacteriophage are the pathogens. Okay, so this guy showed that these were not originating from the bacteria itself, but they came from the foreign genetic elements like the bacteriophages or plasmids. So first there was the repeats identified. Next, people showed that they always were in cluster, and they also associated with some genes called as Cas genes. Next, people showed that these repeats were not the DNA from their own genomic origin, but they came from the foreign genetic elements. So now people knew what these structures were, but still they didn't know what they were doing and why were they there. Then came a paper here on the left. There is, I don't know if you can see my arrow, if you can see that there is a biology direct paper. This was a breakthrough paper and a landmark paper in this, which for the first time suggested that this proteins and the RNA that were found in this cluster of CRISPRs had a similar property like RNA interference. That means most likely they were evolved as immune system against the invading foreign genetic elements like bacteriophage or plasmids. And this was the first paper that hypothesized that this is probably their function. Till then, their function was not hypothesized. Then later on, just 1 year after that, you can see in the other paper, that's the science paper, which says that CRISPRs provides acquired resistance against viruses in prokaryotes. So this was the first paper that showed that, in fact, the CRISPRs that are there, they were there for a purpose. That is, they were the ones that kept the memory of the viruses that infected prokaryotes, and they were utilized to fight against the prokaryotes. This is like our normal immune system. That is, let's say most of us get vaccines, or most of us get once or other time an infection from a virus, but next time we don't get it. Because once we have an infection, the immune system recognizes it, produces antibodies, and it fights against it. At the same time, some of this memory is retained in the immune system in the form of memory B cells or T cells, and next time when you have the infection, these guys will recognize it and fight against the invading pathogen. Similarly, here the CRISPRs contained the DNA or the genetic material from these viruses that attack the prokaryotes. It was kept as a memory and was used to fight against them in the next infections. Okay. So this was the first one to show that probably this is a bacterial immune system against the virus. Then actually the real players, that is Emmanuel Charpentier, comes into picture. Soon after this paper, after nothing much happened between 2007, but around 2011, she came up with a paper where she clearly described the structure of this CRISPR locus, and she identified a very important component of the CRISPR locus called as tracer RNA, which you can see in the bottom. And she showed that this tracer RNA was essential for the maturation of the CRISPR, and without this you cannot have the CRISPRs working against the invading viral DNA. So if you look at this, sorry. Okay, if you look at this, what you can see here is there is a cluster of CRISPRs here, CRISPR 01. You see a box that is rectangles of solid black and rhomboid structures of green. So these are the repeat elements. These are the CRISPR repeats, okay? The same repeats which was observed in 1987. Then beside it, you will see the CSN1, Cas1, Cas2, CSN2. These are all the CRISPR proteins or the Cas proteins. Okay, so they are always together. And just beside that, what you see is the tracer RNA. This is the RNA that is produced, which is utilized by the Cas to mature the CRISPR RNA, which is shown on the right. So this paper showed that all the components of the CRISPR were identified by now. So you add the CRISPR RNA, tracer RNA, and the proteins or the enzymes that are required for CRISPR. But what was not known is how do they work? How all these three things are brought together to fight against the virus. Okay. So if you look at the landmarks, it started at 87 and it picked up pace after 2000 when the cluster was found. And by 2011, the tracer RNA was discovered. So in a conference, Emmanuel Charpentier, she met Jennifer Doudna, who was a structural biologist. So they both decided to collaborate and try to understand how these three components, the Cas proteins, CRISPR RNA, and the tracer RNA came together to fight against the viruses. So this is the paper that was published in 2012 in Science, a programmable dual RNA guided DNA endonuclease in adaptive bacterial immunity. And this cartoon explains exactly how the CRISPRs work. So I would like to spend little time on this. So let's say in a bacteria, whenever there is an infection from a phage or a virus, the DNA is injected or the RNA can be injected, whatever the genetic material is injected, mostly it's the DNA, let's say. Now, the Cas proteins which we talk, which are from the CRISPR locus, they are always expressed in the cell. These Cas proteins, they will break the DNA and take a small part of this DNA and stitch it back into the locus, CRISPR locus, and keep it as a memory. This is a very rare event because when a phage infects bacteria, most of the bacteria die. They will die in millions. But this may happen by chance in one or few bacteria, which will take a part of this DNA and keep it as a memory. Now, they will build a library. If you see here, there are different colored ones. So for different virus, they would have built a library of it. So this acts as a library and the memory for which is an infection, for which infectious agent is specific to this color. Okay. Next time, if it has an infection from such a bacteria, what happens? It will be able to take care of it. Now, how does it take care? Now, as I said, when there is an infection, a part of the DNA is stitched as a library, and if this bacteria survives, the memory is carried forward to its progenies. Later on, what happens? This CRISPR, this is now the CRISPR RNA, which is constantly expressed in the cells. Now forget about bacteria, now forget about the virus, now the bacteria has survived. In this bacteria now, this CRISPR locus will always express constitutively. So this RNA is produced and also the tracer RNA is produced, which is shown in the blue here. Both the tracer RNA and the CRISPR RNA bind to each other. They have a complementary region. Okay. And there are RNase 3, that is a natural enzyme produced inside these bacteria. This RNase 3 processes these hybrids of the tracer RNA and the CRISPR RNA and leads to the maturation of the guide RNAs. And what you see here, the gRNA, it is formed by two RNAs. One is coming from the tracer RNA, other is coming from the cRNA. Okay. Now, this is loaded onto the protein Cas9 in case of Streptococcus pyogenes, and there are different Cas proteins in different species. So we will stick to the example of Cas9, which was shown by Doudna and Emmanuel Charpentier. So now you find a complex of Cas9, tracer RNA, and the cRNA. So it is a 3-component system. Now this will look for all the DNA that is expressed or that is present in the cell. If there is any viral RNA or its own genomic DNA, it will go and open up the DNA and scan for the complementarity of this the guide RNA. So if wherever it finds complete complementarity, that DNA it will cut. Since this DNA was taken from a virus, if a virus infects and the DNA is available in the cell, the Cas9 and the guide RNA complex binds to it and it will try to unwind and see if there is a complementarity. And for this, there is a sequence called PAM, which I will explain next. It is required. which is not there in the viral genome, the bacterial genome. So it has to distinguish between the viral DNA. This sequence is also there in the bacteria. How it distinguishes is through a PAM sequence, which I will talk to you later. But what it does is it unwinds the DNA, looks for complementarity. Wherever it finds complementarity, then this Cas9 has got nuclease activity. It will break the DNA just like a restriction enzyme and creates a double strand break. After that, the DNA will go for degradation. This is how it can specifically degrade the viral RNA or DNA. So this is how the system works. Now, this is understanding of how the CRISPR works. The application of it comes later, where when they decided that if this can be so specific to a viral sequence, can we use it in a mammalian cell, let's say human cell? And if we decide that this is the 20 bases of a gene, can it go there and specifically edit it? And that's the application of it, which I'll be talking in the next slides. So here I will also like to mention one more very important thing which most people would have missed. When Jennifer Doudna and Emmanuel Charpentier were working on this and finding the mechanism, there was another scientist, Virginijus Siksnis, from Lithuania, a small country which many of you may not even have heard of, an erstwhile USSR component. He independently identified the mechanism. He is a person who works on restriction endonucleases, and he independently identified the mechanism. And in fact, he submitted his paper much earlier than Jennifer Doudna and Charpentier to a journal called Cell. And however, they did not take it up and they rejected, and he had to then go into another journal called PNAS. And whereas Jennifer Doudna and Emmanuel Charpentier, they submitted it to Science. Science is another journal which took this important work and they went on a fast track and published it quickly. And Virginijus could publish his work 3 months after that. That's how he lost the priority. If not, we should always respect other scientists working on it and coming up with the same independent findings. So his finding is as good as the finding from these 2 Nobel laureates. So as a recognition, Cavalli Prize was actually awarded to all the 3 of them together. So his contribution is no less for the CRISPR field. So that's something I would like to point out here. So this happens in science, that many scientists will be heading from different directions to the same destination. Some reach early, some reach little late, but nonetheless, their efforts are equally important and valuable. Okay, now this is the history of it. This is the importance of the finding, and the rest of the things you will find the news of it, all those things you can find on the web. Now, since there are students, I would like to try to explain how these things work, what is its importance, and how it is impacting biology. So, coming to the details of the mechanism, as I said, this has got 3 components. One is the Cas9 protein, which here you can see in the pink. Then the guide RNA. Guide RNA has got 2 components. If you see, the green part is the sgRNA component that will give you the specificity to the locus. And then you see an orange loop. That is what is recognized by the, Cas9 to load it onto the Cas9 complex. Okay. Then if you see here, there is a small sequence called PAM, which is depicted in red, that is NGG. Now, PAM is a very important sequence. This is called as protospacer adjacent motif. Now, as I said, whenever there is a viral infection in the bacteria, a part of the viral DNA is taken and stitched into the bacteria. Okay, it is kept in the CRISPR locus. Now, again, when the virus comes, now this will be used as a library to fight against the virus. Now, as I said, this sequence of 20 bases that is kept in the CRISPR locus and in the viral DNA is same. How does the CRISPR system cut only viral DNA but not bacterial DNA? That is because of this PAM sequence. So protospacer adjacent motif is a small sequence of 3 bases in this case, which is present only in the virus but not in the bacteria. So when the virus picks up, when the bacterial CRISPR system picks up the DNA from virus, it will remove this PAM. It is leaving the PAM in the virus itself, and it picks up just 20 bases before it. So that's how it can distinguish between its own DNA and the viral DNA. So when this guide RNA, so what they also showed is, instead of having a tracer RNA and a cRNA separately, you can synthesize them into a single RNA called as single guide RNA, which will also function just like the combination of tracer RNA and cRNA. So henceforth, everybody started using only single guide RNA. You don't need two RNAs. But remember that it has 2 parts. One part is the stem loop, which is essential for recognition of the Cas9, and this green part, that is what is the one that gives you the direction to the specific sequence. Now, the stem loop part is common to any guide RNA against any locus, whereas the green part of the sgRNA is the one that gives you the specificity. Okay, so this CRISPR complex, which contains the guide RNA and the Cas9, will bind to the DNA, and this complex can unwind DNA. So it will unwind and looks for complementarity between this genomic sequence and the guide RNA sequence. Wherever it finds a proper complementarity, then the nucleus domains, one is represented here with a scissor by name HNH, and another is a nucleus domain RUVC, which is also represented by another scissor. So RUVC will cut on one of the strand, HNH will cut on another strand, and then it creates a double strand break. Okay, this normally happens 3 bases before the PAM. Okay, this is for Cas9 CRISPR system, and there are different CRISPRs where it might vary a bit, but this is one of the examples. So this is how it creates a double strand break. And in case of viruses, let's say once the double strand break is created, the DNA goes for degradation, because these loose ends of the DNA are recognized by the endonucleases and they will chop it off. Okay, so this is what CRISPRs do. They don't do anything else. They don't go and change the locus or anything. They actually create a double strand break. Okay. Now, Jennifer Doudna and Charpentier showed that these things can work very well in the test tubes, and they filed the patent. Meanwhile, another group from Harvard, MIT, that's Feng Zhang's group, who is quite popular, he came up with a paper showing for the first time that this system could be used even in mammalian cells directly. So they showed the mechanism, complete mechanism. The Charpentier and Doudna showed complete mechanism in the test tube. He showed in vivo within the mammalian cells this could happen. So you can use this to change the DNA of the mammalian cells. So this, in fact, was a paper that got highlighted, and all the resources of this were freely distributed, and everybody jumped into using this. And that's how Feng Zhang became quite popular. So this was a very important paper that came just a couple of months after Jennifer Doudna and Charpentier's paper. And also, many of you might have read about a lot of controversies around the CRISPR patents. That's between MIT and UC Berkeley. That's because what happened is, although Charpentier and Doudna filed the patent much earlier than Feng Zhang, they left it to the normal course, whereas MIT decided to pay more money to the patents office and get it on a fast track, and they were awarded much early. However, the dispute to certain extent is resolved, to certain extent not resolved. Now, I think Charpentier and Doudna have a priority in Europe, whereas Zhang and MIT have a priority in US for this patent. Okay, now till here I have told how CRISPRs work. Now you don't know how these can be used for manipulating the genome or the so-called genome editing. What does genome editing means, and how it is utilized, and how this has changed the field. Now that's something I'm going to talk the second part. So Genome editing is basically to change the genome as per your desire to achieve a certain objective, whatever that objective could be. Okay. So manipulating the genome of the cells is not so easy, although now it seems to be much easier, but it was a very difficult thing to do. And before the CRISPRs came, it could only be done in very few organisms, some lower organisms like bacteria. In bacteria, you could knock out a gene, you could change a gene much easily, whereas it was not possible in most of the mammalian cells. The only mammalian system where genetic manipulation was possible was only in mouse, because in mouse people had derived something called as germline competent embryonic stem cells, which had very high capability of homologous recombination. I will explain to you these things in the next slides, where at a specific site you could manipulate the genome. And that's why it was only limited to mouse, and for this technology, Nobel Prize was awarded in 2007. And as I said, it is also possible in some of the lower organisms like, okay, let's say bacteria, some of the bacteria, yeast, where it is possible to do homologous recombination and change the genome specifically at a specific site. However, in whichever those species where it is possible, it was a very elaborate process, very low efficiency process, and also involves a lot of expenses. So it was not often employed regularly. Then parallelly, when this CRISPR field was developing, where the CRISPR field's idea was not to devise something to do the genome editing. They were only studying a phenomenon, and then they came up how that can be used for genome editing. Whereas there was another set of researchers who were trying to work and create nucleases which can specifically manipulate genome or edit the genome. In this process, people discovered something called as zinc finger nucleases and TALENS, which were better and became workable in many species, but were still difficult to adopt and set up because they were either difficult to build them or use them, or they were also very expensive. Okay, so CRISPRs basically brought two fields together. Okay, so one field was coming parallelly where people wanted to manipulate the genome and they were trying to find methods. And on the other side, the bacteriologists were trying to study an immune system against viruses. And later on, when the mechanism came out, they realized that this could be used in mammalian cells or any other cells to manipulate the genome. So that's an interesting lateral flow of thoughts and ideas. So that's something you should appreciate here. Okay. Now, how was the genome manipulated in, as I said, in some of the species it is possible. That was by homologous recombination. So homologous recombination, you might have read, this is something that happens during meiosis, but it also happens in other cells. When you provide a DNA from outside, this can happen, but this happens at a very, very, very low frequency. So let's say in a bacteria, if you want to target a gene or insert a gene or knock out a gene specifically at a specific site, let us say, this is the example I would like to show here. Then what you do is you pick up the DNA sequences just around that gene. For example, I want you to focus. I hope you can see my arrow here. If this is a targeting vector that is depicted here, most of you might have come across it in your textbooks. Let us say this pale orange structure is the gene which is shown here, which I want to disrupt, or I want to introduce a selection cassette there, specifically there. I don't want this cassette to go and get incorporated anywhere else in the genome, only in that space. Then what one can do is take a selection cassette like neomycin or something and put homologous regions on either side of this neomycin cassette, which is specific to that site. Now, this part is a left homology region and a right homology region corresponding to this gene. When you put this into the cell, this DNA can have two fates. This DNA can just go and randomly sit somewhere, which is shown on the right. This is called random integration. This is not a specific event. So it's a random event. It's a transgenic. So here you did not achieve specifically going into a locus. So This is not a desired event. Whereas sometimes what happens, because of this long DNA homology regions, the DNA repair systems will recognize the end of this DNA and they will try to stitch it into the specific locus. This happens at different frequencies in different cell types. In bacteria, it happens at a little decent frequency. In case of mouse embryonic stem cells, it happens 1 in 10 raised to 6. That is the sort of a rare event this is. Okay. And in some cases, in somatic cells, it is much low. So you can practically pick up such events. So this happens because of the homology regions in these two parts. This leads to homologous recombination. You have learnt about the Tolley junction and all those things, crossover, non-crossover, ultimately leading to the stitching of this targeting vector which you gave into the locus. This is a homologous recombination event which happens at specific site. Okay. Now, how do CRISPRs help in this? So this is the same technology that was used in the knockout mice, where you can create this one targeting vector, put it into the ES cells, and you manipulate the gene. Then you inject the cells into the blastocyst and create a transgenic mice. Okay. But as I said, homologous recombination frequency is very, very minuscule, and it mostly doesn't happen in many of the species. So it's very difficult to get this working. How to enhance this? If you can enhance homologous recombination, then you can go to the specific site and repair it or change it the way you want. So this is possible when you can create a double strand break in a specific locus. Now remember, CRISPRs I was telling can create a specific double strand break. So if you can create a double strand break, that will recruit the DNA repair system, and that can change the locus very easily. So here are 3 different possibilities when a double strand break happens. So basically, CRISPRs create a double strand break. Once, or let us say, there are many means by which you can create a double strand break, by radiation, by other mutagens. Once there is a double strand break, there are 3 possibilities the DNA will take. One, it can, most of the times, it will go through a pathway called as non-homology end joining, where the DNA are stitched together back. But while they are stitching this DNA back, either few bases are lost or few bases are added. So it is not a specific, or you don't know the outcome of the event. There could be few bases added, which is called insertion, or few bases deleted, which is called deletion. So there is a change in that locus. The other thing that happens is, when you have a double strand break, and in that cell at that time, if you have given a donor template, mean to say a template of DNA, double strand or single strand, which has got homologous regions on either side of this break, and with whatever the sequence in between, that will be stitched in. This is homology dependent repair, and this is exactly where you can get a precise alteration of whatever you are thinking. You can put small changes, you can incorporate small changes, or you can remove small changes. So that is possible. Okay. And this is what is the crucial point. So there is, you can give a different donor template and there will be precision, this one that happens. Okay, the precision incorporation of this. So when there is a double strand break in a cell, there are 2 pathways you should appreciate. One is non-homology end joining, which is not specific or not precise. Whereas when you give a donor template, you can achieve a specific change that is called homology dependent repair. And this is what can give you a proper editing of the genome. You can change the locus the way you want. Now, if you see here, there is no CRISPR, nothing is involved here. This is what the cell does. So what does CRISPR do? CRISPR does anything before this, that is to create the break. That's it. That is the job of the CRISPR. After that, what happens is dependent on what condition the cell is in. You don't give any donor template, it will go for non-homology end joining, where you have a site-specific change, but you don't know the outcome of the change. Whereas in case of homology dependent repair, you can get a site-specific change of your choice. Okay, that's what it is. So as I said, double strand breaks can create this or increase the efficiencies of changing a specific locus. So as I said, parallelly people were developing different technologies, and zinc finger nucleases was one of them, where zinc finger motifs are present in many of the DNA binding proteins. So people developed or engineered different zinc fingers which can recognize a specific sequence of DNA so that you can locate it into a specific locus and cut that. Now, these are called site-specific nucleases. Now, CRISPR, zinc fingers, and TALENS, all the three are site-specific nucleases. Now, what is their importance? I will tell you. If you see the genome, it's in case of mammalian genome, it is 3 GB. Now, if you, in that huge 3 GB genome background, if you have to locate to a specific DNA sequence of 20 bases, that means you have to have it going there. It is like a target seeking missile. That's one of the examples people use. And I use an example of a simple example of a postcard. So let us say India is a huge country with 140 crore people, nearly 140 crore. If I have to send a postcard to a particular person, let's say in Mysore University, let's say some arbitrary name we will take. Let's say we will take Manjunath, one of the names. If I write on the letter saying that Manjunath, now in the world it can go to anybody. This is like a restriction endonuclease. Because restriction endonuclease can recognize 4 bases or 6 bases, it can cut in multiple places in your genome. Okay, so if I write only the name, there will be thousands and lakhs of people with that name. Similarly, restriction in the endonucleases cut at multiple sites in the genome. But you want to target only one site. That means now you have to go more specific. Then if you write the name of the person, the country name, state name, you are increasing the specificity. So likewise, if you keep going down to the extent that then you write the name of the state, then the city name, road name, and the house number, now that letter will reach exactly the person where it should have reached. So that is exactly what it is. Normally 20 bases in a genome, it's 18 bases if you go and compute it in a 3 GB genome. 18 bases can give you a very specific site, a single site in the genome, or 20 bases. So that's why these were engineered to recognize around 20 bases so that they go only to one locus, not to other locus in the genome. You can change that one part and go and create a double strand break there. So people created zinc finger nucleases, which were very difficult to design, and also there were patent issues, very difficult to design. Mostly people used to buy them. They were not very popular among everybody because it was very difficult to create them. And the knowledge of which zinc finger motif recognizes which sequence was also not very well available. These things were developed in 1990s and became, their application started in early 2000. So some of the companies used to provide services for this. And in case of zinc fingers, a zinc finger single motif can recognize around 3 bases, and you can have multiples of zinc fingers to recognize 18 or 21 bases, and you can put a FOC1 restrict endonuclease sequence or the domain which will cut the DNA. So the zinc fingers in this case, the zinc finger domains will give the recognition to the DNA, whereas the double strand break was created by a FOC1 endonuclease domain. Later on, another system came called as transcription activator-like effector nucleases. This comes from a bacteria called Xanthomonas. Here what happens, an amino acid sequence of 34 amino acids, which is shown here, with the 13th and the 12th and the 13th amino acid could recognize one base. Let's say in this 12th and 13th place, if there is HD, it can recognize C. If you have one more repeat of this, and if it has NG in this 12 and 13, it can recognize T. If it has NI, it can recognize A. So if you have a 20 bases sequence of DNA, by having these 34 amino acid peptide one by one stitched in tandem to each other, you will be able to recognize that 20 base pair sequence. So a specific TALEN can be created for a specific DNA sequence. So in both the cases, be it zinc fingers or TALENS, you have to, if you want to target a specific sequence in the DNA, you have to create a protein that is specifically binding to that. So basically, in this case, the DNA sequence is recognized by the protein. So the direct interaction of the protein with the DNA will give you the specificity. So likewise, people were able to, this came around in 2010 and TALENS became really popular. They were much easier than zinc fingers. Many people could actually use them in their labs and could go into specific sites and create a double strand break. If CRISPRs hadn't come by 2012, if they were delayed for another 5-6 years, let us say, TALENS would have become actually very popular and most of the labs in the world would have been using actually TALENS. It's just the CRISPRs came and TALENS went out of fashion, because TALENS, although were easy to design, were still like for every DNA sequence you had to create a new protein. Whereas in CRISPRs, as I said, it is the RNA that recognizes the DNA sequence. So the Cas protein is common, irrespective of which gene or which locus you are targeting. You just change the RNA sequence, that's it. Whereas in case of TALENS and zinc fingers, you have to change the protein itself for every DNA sequence you want to recognize. Okay, so TALENS were the next ones to come. And in fact, they became very popular. And once the CRISPRs came, they went out of fashion because CRISPRs were very, very easy to use. Now, as I said in the previous, the protein sequence will recognize the DNA directly. In case of CRISPRs, as I said, the Cas9 remains the same, irrespective of which gene, which locus you want to target. Even in the sgRNA, the stem loop structure here remains the same. It is only this cRNA region, which is 20 bases, that is always changed depending on the target where you want to target. So CRISPRs also do the same thing. They go and create a double strand break. So be it zinc fingers, TALENS or CRISPRs, everything creates a double strand break. After that, what happens? As I said, non-homologous end joining leads to either deletion or addition of DNA, whereas homology dependent repair can specifically put the given DNA in that locus and change that locus. Now, what this has done is, the homologous recombination, as I said, is a very inefficient process. But when you create a double strand break, the DNA repair system gets activated. And if you provide the donor DNA, it works more efficiently. So CRISPRs only create a double strand break at the required sequence. Rest of the things are taken care by the cell's repair system. So CRISPRs end actually here, or any nuclease, site-specific nuclease function actually ends here after creating a double strand break. After that, the cell's internal machinery will take care of it. Okay. Now, after CRISPRs, after the Cas9, I said in case of Cas9, it required 3 components: a Cas9, a tracer RNA, and a cRNA. Later on, after a couple of years, people came up with another Cas system called CPF1. So above here, you can see the Cas9 system, where you have, this is a type 2 system, where you have the CRISPR locus with Cas protein, Cas9 protein, and the tracer RNA. Whereas in type 4 CRISPRs, which is CPF1, example is CPF1, you have a CRISPR RNA here, and only Cas protein, that is CPF1 protein. This doesn't require a tracer RNA. So in this case, you just require only one RNA, no 2 RNAs. And it works little differently. I told you the protospacer adjacent motif, PAM. In case of Cas9, it is MGG. So this works very well. The Cas9, that is CRISPR Cas9, which is shown on the right side, works very well where the genome is GC rich. Whereas in those genomes where it is AT rich, CPF1 works well. And the difference between these two systems also extends further. In case of Cas9, you create a double strand break, which is a blunt break. Whereas in case of CPF1, the cut is little away from each other. So it creates a sticky end, and also the cut is on the 3' end of the recognition sequence, not on the 5'. Okay. So there are multiple Cas systems that have come and multiple CRISPR systems that have come. These are just few of the examples. Now there are actually hundreds of them that are coming and from different bacteria, and they have been utilized in the similar fashion. Now, what are the possibilities with this sort of a CRISPR system or a site-specific nuclease? What people have been able to achieve and where they have used, and how much manipulation of the genome can be carried out. In fact, you can do literally anything you think in the genome. Examples here are, you can add or delete a transcription factor binding site in a gene, or you can delete the promoter of the gene, mutate the promoter, add a new promoter, create a new promoter, or introduce a new constitutive or an inducible promoter. You can knock out the gene, you can disrupt the gene by deleting a part of the exon. You can make point mutations. Now, point mutations are very, very important. Particularly, I'll be talking about its application in monogenic disorders where a single mutation creates a disease. These become very important. You can go and correct the mutations. You can fuse a reporter gene. You can create safe harbor sites. I don't want to explain those. You can create knockouts, reporters, many of these things. So basically, you can manipulate the DNA the way you want within the cell. Okay, that is something that was not easily possible before. Now it is easily possible in any type of cell line, in almost all type of model organisms. Okay, almost all model organisms, you can manipulate the genome using CRISPRs or even TALENS. Okay, so these are some of the earliest findings. You can not just target one sequence, you can target multiple sequences. You can create mutations in different chromosomes simultaneously at a time. So you can multiplex the genome editing using CRISPRs. Okay, and there are many other utilities I'm talking about there. So I said in the Cas9, there are two domains we talked about. One is the RUVC, which is a nucleus domain, and the other is the HNS. So each Cas9 has two nucleus domains. One nucleus domain cuts in one strand, the other nucleus domain cuts in the other strand. You can just make point mutations in these domains and you can kill the nuclease activity. These are called as dead Cas9. Okay, so does it affect the ability of the Cas9 to form the complex and bind to the DNA? No. This in no way affects the ability of Cas9 to interact with sgRNA, go to a specific locus and bind to the DNA and open it. It does all the function except that after opening the DNA, it can't just cut it. That's it. Otherwise, a dead Cas9 functions exactly like a normal Cas9, except it can't create a double strand break. So what people did is, since a Cas9 and a guide RNA can go to one specific locus in the genome, can we use it to manipulate the genome in different ways? So what people thought is, if I design a guide RNA which goes specifically to the promoter of a gene, either I can switch on the gene or switch off the gene. So there came systems called as transcriptional activation, CRISPR-on systems, or transcriptional repression. Here is an example of transcriptional repression. I am circling it here, where you have a dead Cas9, dCas9 is a dead Cas9, there is a guide RNA, it will go to the specific locus. Let us say it is going to promoter of a gene X, it go to that promoter and binds there. And this Cas9 now is fused to a crab domain, which is a repressor. It can bring transcription repressors and repress this gene. So if you want to shut down a gene in a cell or in an animal without changing the DNA sequence, you can use CRISPRs to shut down the transcription of the gene. Okay. Similarly, a gene is switched off, you want to switch it on. Same Cas9, dead Cas9 can be used, except that instead of crab domain, now you have an activation domain like VP16, which brings transcription activators, and this will lead to activation of the same gene. So you can create, using the same CRISPR system, an activation system or a repressive system, or you can target a gene and image that specific gene in the locus. In the whole whole genome, you can go to a specific locus, and if you fuse some fluorescent proteins to Cas9, you can image that specific one. Or you can have photoactivatable, transactivatable CRISPRs as shown here. Or you can have something that will manipulate the genome. For example, epigenetic modulation of genome also can be done. To the same Cas9, you can add a demethylase or a methylation enzyme, and it can, wherever it goes, it can create the methylation or demethylation or acetylation. So you can basically manipulate the genome, manipulate the expression of the genes, or even create epigenetic manipulations without changing the DNA sequence. So these things are all possible, and these are few examples. There are multiple uses of CRISPRs. In fact, you can't imagine. Whatever you can think of, most of the modifications can be carried out using CRISPRs. So later on in the past couple of years, in the past 3-4 years, people started thinking, because although CRISPRs are known to be very specific to a locus, sometimes they can also at low frequency go and cut some other sequences. So this creates an unwanted cut. So people started thinking of ways to change the DNA without cutting it. Then came something called as base editors. So base editors basically contains the same dead CRISPR system or the dead Cas9 system, except in the previous ones I said they have tagged the Cas9 with either transactivation domain or the repressive domain. Here you can either tag it to cytidine deaminase, which is shown here. When the CRISPR system binds here, the Cas9 binds here, this cytidine here is exposed to the cytidine deaminase tagged to the Cas9, and it will deaminate it, which later on in the subsequent replications will be converted to T. So a C to T change can be brought about without cutting the DNA. So you can make point mutations without even cutting the DNA. Similarly, a to G can be brought about using another base editor called adenosine deaminase. Okay, so basically you can use CRISPRs to create point mutations, one or multiple, even without cutting the DNA. So its applications, particularly in correcting the monogenetic disorders, is immense. Not only DNA, you can even edit the RNA after the RNA is transcribed. Here a different Cas system is used, Cas13b, even Cas9 has been, because Cas13b actually recognizes RNA instead of DNA. Whereas there are also Cas9 systems which can even edit the RNA. So you can even change the RNA also. So basically, what CRISPRs have done is they have given us a tool to specifically go to a particular locus in the genome and change it as per our will. Now this is amazing, because previously such sort of things could rarely be done and could only be done in very, very few organisms. Now it can be done in almost any organism you can think. So genome editing has been demonstrated in almost all the model organisms. Particularly of interest is the crop species. Most of these crops which we see, its application in agriculture is huge, and most of these crop species we see there, the genome editing is demonstrated, and people have shown that they can change the genome, they can delete, add, they can change the bases, all these things. And the best part about CRISPRs, why it became so popular, is this is an open access system. That means all the resources that are required for setting up a CRISPR were available openly to everyone. There was no restriction, whereas this restriction was heavily found in case of zinc fingers, whereas these were available to everyone. For example, Feng Zhang's paper came in February 2013 in Science, and by March, all the plasmids used by him were available in AdGen for purchase at a very nominal price by anybody in any corner of the world. And also, the people didn't have to worry about how to design a guide RNA for my specific gene. There were softwares and programs that were made by multiple groups and were put out in the public domain, and people could easily go in, put in their sequence and design the guide RNA for them. So this democratized genetic manipulation. It democratized in a huge way. It was made accessible to everyone and to every model organism. So this has changed the space of biological research, and it has opened up all those new possibilities and to ask new questions which were before unthinkable, because the scale of modifications that can be possible in the genome now are immense, and previously what people wouldn't even imagine, people are able to. And commercial implications, as I said, are very immense, because in agriculture there have been already a lot of crops that have been developed. there have been many disease-resistant animals that have been created, viral-resistant. For example, let's say if a virus needs a receptor to enter a cell, the mutation in that receptor will prevent the virus from entering it, and you can create a strain of a or a breed of a animal which is resistant to that virus. And many actually the pig in the pig, lot of strains of animals have been created which are resistant to many of the viral diseases. And also some of the gene therapies, particularly the monogenetic disorders of human origin, particularly the hematological disorders, are in clinical trials now. So with this, I would stop we come to the end of this talk, and I would like to acknowledge that all the images are sourced from either the Nobel website or from the internet. And in the end, I would like to leave a word of caution, that is, this is a very powerful technology that needs to be handled with responsibility. And I think most of the countries across the globe are building in proper ethical pipelines and regulatory mechanisms to handle this, because these are very easy to set up and use in the labs. Okay, so here I end, and I will be happy to answer any questions you guys have. Are we still online?
Thank you, sir. It was a very compelling and enthralling experience to hear from you. Now the topic is open for discussion. Put it in the chat box.
Yeah. Sir, I am Dr. Girish Naik. Sir, I congratulate you for a very exhaustive and excellent presentation. Sir, do you have an example of successful application of CRISPR in crop improvement? Any crop?
Actually, many things have not come into the market. In fact, the biggest utility of CRISPRs has been in agriculture. DuPont had a major research section in Hyderabad, which is now shut down. They created multiple varieties of wheat, particularly on this, particularly the ones, I don't exactly know the traits, but they have created a lot of them, particularly which involved either removal of a gene, because those were the easiest to create. Yeah. And they have engineered a lot of them. And across India, there are multiple places where these have been done in most agriculture universities. In fact, that's a place where it is utilized heavily.
Yes. Thank you, sir.
Sir, we have one question from Anuj Bandral.
Yeah.
Is it possible to use CRISPR-Cas in the case of new organism or newly found organisms? If yes, then how to proceed with it?
So to use a CRISPR-Cas9, so what you have to do is you will have to design a guide RNA. Let us say I want to target gene A in a new organism. I need to know the sequence of that gene. Yes, it is possible to use it in a new organism, particularly if the organism has got a smaller genome. It is very easy to sequence these days. You can sequence it. If you have the sequence of that organism for whichever locus you want to design a guide RNA, it is easy to design. So to create a CRISPR-Cas9 for any organism, you need to have the genomic information. That is must. And the Cas9 functions in almost all the organisms. So that's not a problem.
One more question is from Meghna J. In cells co-transfected with a pair of sgRNAs to mediate and edit in the DNA, how can the indel mutations be detected? How is the accuracy checked?
Okay. So when we, as I said, when we use CRISPRs, we know the sequence of the genes which we are dealing. Let us say you targeted a gene A, and the Cas9 went and cut the DNA. Now you want to know what is exactly changed there. Has it changed a few bases, multiple bases? Are there any insertions or not? So wherever you are cutting, you use 2 primers on either side of that cut site, amplify the DNA. Then simplest thing to do is just send it for Sanger sequencing. You will get the results in 2 days. You will be able to precisely say which base is missing, which base is added, everything. Previously, people used to use something called as surveyor assay and other things, but sequencing is the simplest, where you can precisely say which base is removed, which base is not removed. So whichever target you cut, amplify that sequence and send it for sequencing. You will know it.
Thank you, sir. There is one more question. What affects the rate of RNA mutation? And the second part is why the more number of nucleotides or gene makes RNA more stable?
Okay, this is tangential to what I am. So what affects the rate of RNA mutation? So I don't exactly understand it. Mostly the mutations we talk are in DNA. Of course, there can be transcriptional errors which can be incorporated, and that creates a different sequence of RNA. But RNA also gets a lot of processing after it is made. There are modifications to RNA and there are base modifications that happen. So I don't exactly get this question. The rate of RNA, most of the times the rate of mutations we talk is with respect to the DNA. Why more number of nucleotides or genes makes RNA more stable? So RNA stability is, okay, this is also a bit of a confusing question, but RNA stability is dependent on many things. Okay, sequence is one of them. The secondary structure and its association with other proteins which forms RNP. So these are all the ones which determine the stability of RNA in a cell. So there is not one process, but there are multiple processes. Of course, the secondary structure is one of the primary one.
Ms. Sharma, if you are there, you can unmute and get more insights to discussion.
Hello.
Yeah.
Good afternoon, sir.
Yeah, please.
Sir, I was watching a video as there was a project of coronavirus I was doing. So, in one video, it is said that the number of genes in coronavirus makes the RNA virus more stable.
Okay, now I can understand the context. So the context is totally different. So the size of the RNA and the modifications, even the viral RNA gets modified. Some of them have poly-A, some of them have caps. So the size and probably its packing, that makes a difference. Okay, with respect to the RNA viruses. Okay. And the rate of mutation is dependent on certain things. The more number of bases you have, the probability of mutations are high. And also it depends on the replication rates. And most of the times the replication of these RNA viruses are within the cell. It also depends on the proofreading capability of the cell in which it is replicating. So the mutation rates are dependent on multiple things.
Okay, sir. Thank you.
Are there any more questions?
Looks like there are not any more.
Okay. So if there are no more questions, then we thank Dr. P. Chandrashekar for his lecture. And now we have come to the end of the morning session of lectures. We will have a break of 1 hour till 2:30 p.m. and rejoin to hear the third lecture for the Nobel Prize 2020 in the field of physics. Thank you.